Welcome to Hustle, Style and Stories with Shanu Suresh. We often hear the term gene editing. Now, what exactly is gene editing? Can we really mitigate serious health conditions by simply editing our genes? And a very important next question, is it safe? So today on the show, we have the right person to answer all these questions and talk about all this and more. He is the CEO and co-founder of Mammoth Biosciences, Dr. Trevor Martin. He is on a mission to make humanity and make the lives of human beings better by simply getting rid of serious health conditions. And not just that, being a scientist is one thing. Creating waves in the world of science, healthcare, technology, biotechnology is one thing. But also successfully scaling a business, that is another. He has raised funds to the tune of $400 million plus and will continue to rise. Let's talk to Dr. Trevor Martin on the show today at Hustle, Style and Stories with Shanu Suresh. Welcome to yet another exciting episode of Hustle, Style and Stories with Shanu Suresh. Well, there's so much more that goes on at Mammoth Biosciences. I have the co-founder and CEO. Thank you so much, Trevor. For doing this for us. Trevor, honestly, let me just jump right in and start by asking you, gene editing. Explain to us like we are first graders. Okay. Of course, I understand a bit of it because I love biology as a subject myself. But I would love to know from you, what exactly is gene editing? What exactly does Mammoth do?
Yeah. So kind of zooming out, the way we usually think about medicine is that we think about treating basically the symptoms of disease. And the power of gene editing is that it allows us to really build what we think of as actually cures for disease. So to give some examples, there's diseases like Alzheimer's or Parkinson's or muscular dystrophies. And these are things where even from birth, by looking at your DNA, so like the code of your life that's in all of your cells, you can tell that a person is either at high risk of or going to get one of these diseases. These genetic diseases is what we call them. And that means that if we were able to change the DNA, we could actually get rid of that risk and make them not have that genetic disease, or even if they already have it, potentially cure that genetic disease. And that's a very powerful concept because that's a very different way of thinking about medicine of, oh, okay, you're going to get the disease and we have to deal with it somehow and deal with the consequences of that, to, oh, actually, let's make it so you never get that disease or even reverse the course of it. And that's really a powerful idea. And it's been something that's the holy grail kind of genetic medicine for decades, but it hasn't been possible because it's very hard to change
to even
the DNA.
Identify the wrong gene in it.
Yeah. So first you have to figure out what in your DNA is causing that disease. And the good news there is that there's about 4,000 genetic diseases where we actually understand that pretty well. There's one location in the genome that very clearly causes the disease. So out of all 2 billion base pairs, you can think of your genome as this big Word document that has 2 billion letters in it. And it's actually a very simple document in some ways because there's only 4 letters, A, T, C, and G, which are the building blocks of life, DNA. But in other ways, it's a very complex document because it's a bunch of A, T, C's, and G's, and you don't know kind of what's causing what. But over a lot of work by a lot of scientists, there's 4,000 genetic diseases where we really understand. So for example, Huntington's disease. This is a devastating disease where basically patients pass away in their 30s due to the buildup of protein. And there's really nothing we can do about it. But we understand what's causing it in the genome. So if we could go into the genome and just change some of those letters, we could actually
mitigate?
Well, not just mitigate, but allow them to live a normal, healthy life.
Okay, so because a genuine question that popped up when you said that, because a lot of these diseases are hereditary, right? So if we mitigate these gene markers or the faulty genes, so you're saying we can actually also stop the progression of the disease in the forthcoming generations in our family?
Well, that's an interesting question. And it brings up a good point that we often think about on the ethical side. So when I talk about gene editing, there's 2 types of gene editing. And the type Mammoth works on, and really all companies work on, is what we call somatic gene editing. Yeah. And that means that if I give you the therapy, you're being cured, but your kids will have the same risk of the disease because it's only in the non-egg and sperm cells. But you can
Oh.
imagine doing editing also in the germline cells is what we call it. So we call it germline editing. And that's a
So
practice
it's
where there's
semantics
more,
and germline.
yeah, somatic and then germline editing.
Okay.
And all companies work on somatic because that's something where there seems to be a general consensus that it's a strictly good thing to cure these diseases.
You end it with the person.
And it's something where their kids have the same just kind of the natural risk of the disease.
Right.
But you can imagine there's a big kind of debate about should you do germline editing and should you have that kind of risk in the children? And I think that's something everyone's going to have like a personal answer for. But as companies, everyone just does the somatic, so everyone can kind of make their own choice.
Is it a very difficult process compared to semantics, or?
Yeah, so in some ways, it is a very complicated process. There's pluses and minuses to the technical sides of each one. From the somatic side, the real challenge is because, let's say you're an adult and you're living with a disease, you have a lot of cells in your body. And that means you have to, somehow deliver the technology to the right cells, like the brain or the heart and muscle, and that can be a challenge. And then B, you have to edit enough of the cells that you're having a real effect, right? If you just edit one cell in the brain, probably not going to do much. But if you edit like 60% of the brain cells, you'll probably be fine. You probably don't need to do 100. Whereas for editing like egg cells or sperm cells, there's obviously less cells to edit. There's other challenges to that beyond the ethical considerations, but that's where the somatic editing can be very challenging, but also it's very important because, there's 8 billion people alive today with disease, and you don't want to say, hey, like we're just going to focus on people that are yet to be born.
Okay.
So I think
you want to solve the problem at hand right now rather than look at the future.
And that's really the low-hanging fruit in terms of there's many, many people that are either living with or will be living with these diseases, and we can actually do something for them.
Fair. So tell us about the CRISPR technology. I know CRISPR stands for a really long, difficult to pronounce. It's an acronym for that. But okay, let's do it. Let's start off by pronouncing what exactly CRISPR stands for, and tell me the process very easily. Like, talk to us like we're first graders.
So CRISPR is an acronym. It stands for Clustered Regulatory Interspace Short Palindromic Repeats. And the reason why it has that wacky name is that the CRISPR technology actually comes out of the adaptive immune systems of bacteria. So similar to how you and I have an immune system, and when we get a virus, it kind of fights back against it, and we get a fever, and we fight off the disease. It turns out microorganisms, like single-cell organisms like bacteria, also have an immune system, and they also fight off viruses. Like viruses try and invade bacteria and turn them into little factories to make more virus. And bacteria have evolved different immune systems, including CRISPR. And part of that immune process with CRISPR is that they actually store a piece of the virus and say, hey, I want to monitor, and if I'm ever invaded by that again, I want to have a very quick response.
Okay, so this is practically the theory that the COVID vaccines also employed, right? You use a bit of the virus to fight the virus.
Yeah, so it's a very common
protein.
It's a very common trend, I think, in science in general, but biology in particular, where you leverage billions of years of evolution, and nature is very clever, right? And it's a very ruthless competition. So there's all these like very powerful mechanisms that have kind of naturally evolved that we can then co-opt and kind of rip out of nature, do a ton of engineering to get it to actually work, and turn into these amazing technologies like CRISPR, where originally it evolved in bacteria as a way for the bacteria to chop up viruses. And we can take that fundamental technology, rip it out, and then use it as a way to cut human DNA in very targeted ways so that we can actually cure disease. So that's a pretty common story, I think, in biology, where you leverage natural evolution, you do a ton of engineering, and you can create these amazing technologies that allow us to actually transform people's lives.
So it sounds very fascinating, all of this. What made you get into it? How did this start for you?
Yeah, so I did a PhD in biology over at Stanford, and I was very fortunate in founding the company to team up with a bunch of PhD scientists from Berkeley, including Jennifer Doudna, who won the Nobel Prize for actually inventing this CRISPR technology that we use. And the genesis of the company was really this fundamental idea that the promise of genetic medicine is that we should be able to edit any cell in your body in any way that we want. And going back to your other question about kind of what does CRISPR actually do? So if we think of your genome as this giant Word document with 2 billion base pairs, what kind of the original use of CRISPR is that you can delete a sentence. It's very powerful at doing that. And for many diseases, that's great. That's exactly what you want to do. There's some sentence that's kind of wrong in the document. You just need to get rid of it. But there's other diseases where maybe there's a whole paragraph missing and you need to insert a paragraph. And there's some where maybe a single word is misspelled and you just need to kind of spell check it. Or maybe you need to bold it or italicize. You can really abuse the metaphor.
Yeah.
But these are all kind of analogs for what you're trying to do to the genome. And it means there's many different types of edits that you need to do. So our idea was you need to be able to do all those types of edits and you need to be able to do it in any cell in your body. And that's where we said, okay, we need to found Mammoth to really deliver on that long-term promise of the technology. Because the technology before that has had a huge impact, and there's actually an approved therapy using CRISPR for diseases like sickle cell and beta thalassemia in the US. But it's been really limited to a couple tissues like blood and liver for technical reasons, because they're easier to deliver to.
Yeah.
Like blood, you can actually take the blood out of the body, do what you want to, and put it back in. And that's awesome for those patients. And these are actually often underrepresented diseases. Like sickle cell is a disease where there hadn't been much focus historically. So it's amazing for those patients. But the promise is going in the brain, going in the muscle, going anywhere, not just blood and liver. So that was really the impetus of like, we need to further develop this technology with new versions of CRISPR, new versions of types of edits. And that's what we're doing in Mammoth.
I have so many questions. Okay, so it's an interesting technology, right? So at CRISPR, what are some of the diseases that you can easily target or have been easy to cure, as opposed to diseases that you're still working really hard on because that's very difficult to crack?
Yeah, so it goes back to this idea of kind of what are the most accessible tissues. So for blood, through, you're probably familiar with like dialysis, you can take blood out of the body. So for those, once you have cells outside of the body, there's a lot of different techniques that you can use to edit them and to kind of deliver the editing machinery. But if it's your brain or your muscle, you can't really rip it out and then put it back in. That would not be a pleasant experience. So that means that the ideal kind of way that you do it is you give the patient a simple injection, and then the technology travels to the right tissue, does the edit, and then safely goes away. But that's a way harder thing to do. The great thing about the technology is you don't necessarily have to edit every single cell. Okay. For many diseases, if you just edit even half the cells, you're going to rescue the disease and the patient won't have it.
Right.
There's some diseases where maybe you do need to edit every cell, but actually you probably don't want to go after those first because those are going to be really hard. That means the technical bar is super high. But thankfully, most diseases are not like that. Because often it'll be some sort of like loss of function. And that means if you can just rescue the function enough, not fully, but enough, then the disease goes away. Maybe some proteins lacking or something like that.
So CRISPR is basically a protein that you sort of insert.
The drug itself that you'd be injecting, what you'd be injecting into the patient is probably some sort of delivery vehicle, like an LNP or an AAV. These are just different kind of containers that can hold some sort of payload.
Yeah.
And that payload would be the CRISPR technology. So then the delivery vehicle goes to the right cells, delivers the CRISPR technology into them, and then that protein binds to the DNA and makes a very specific edit and then goes away. Because you don't need it to stick around in your body because it's just kind of a one and done therapy in the sense of it just changes the DNA. And then even if that cell splits or does other things, it's still going to have that change to the DNA. So it's a very simple concept.
How long does it typically take for a patient for prognosis once this is done?
Well, that's the kind of magical part of it is that really within weeks, you can see the kind of material change in the cells. It's a very fast process in that way. So from the patient perspective, really what we're shooting for is you kind of come into the doctor's office, you get an injection, and definitely a month later, you're cured of the disease.
A month?
Yeah, like it's a very...
Has there been relapses?
So that's definitely the right question to ask. So what we can do now is we can look at the therapies in blood and liver, which are kind of the first ones because of the ease of delivery. And so far for those, we've seen no relapses. Like really,
like
it's
a 100%
effective.
Success rate.
Not only is it effective, but it's also safe because that's the other reasonable question you could ask is like, okay, are you really making this only the specific edit that you think you're making? Or are you kind of doing other stuff? And on both accounts, the really exciting, and these are very reasonable bars to set, but the exciting news is so far, and some of these patients have been followed for years now, we're seeing that it's A, extraordinarily safe, and B, extraordinarily effective. And that's amazing. Yeah, I think there was a world where, maybe that's not the case, but it turns out the technology is actually very, very robust.
So we have a robust product with us. We have a lot of patients that need help. Is it very expensive?
Yeah, so that's another good question as well, because when you have these permanent cures, it definitely saves the hospital system and the healthcare system tons of money over the lifetime of the patient. You might save the healthcare system $20,000, $30 million because these patients have to be high users of the healthcare system to stay alive or to have a quality of life. So often these therapies also can be complex to manufacture because they're new. And there's not many CRISPR therapies out there, so we haven't learned how to bring down the cost of manufacturing. So these therapies often can be very expensive. They can be millions of dollars, actually.
Wow.
But that's a millions of dollars cost that's one time that A, solves the patient's quality of life or allows them to live, and B, then saves the healthcare system orders of magnitude more money of the long run. But it is a very different model. We're used to the healthcare system kind of spending lots of money, but over a period of time. And here you're saying, hey, you're going to spend a bunch of money, but it's going to be less than you spend.
Not a lot of people have the kind of capital to spend.
Usually it's going to be your insurance company
yes.
in the United States.
So somewhere do you have like an ethical argument in your head? It's only for the wealthy to stay healthy?
No, I think it's critical that this isn't something that's just for the wealthy. And I think that's where, insurance companies are not doing this out of the goodness of their heart. Regardless of the wealth of the individual, you have to have insurance generally in the United States.
Most of the insurance, do they cover something like this?
Often they do. And for these therapies, it really is a cost argument of, hey, you're paying more upfront now, but you're going to save a ton of money. It's not altruistic. But it really is a powerful economic argument that you really are saving money, but that is predicated on it has to actually be a one and done. They can't relapse, and it has to be safe so you don't have complications. But so far, both of those seem to be very true.
We've got a bit of lowdown about what CRISPR does, but now let's talk about this from the business point of view. You have raised a lot of money successfully. Congratulations on that.
Yeah.
I think about $400 million and plus and counting.
Yeah.
Tell us about that. What was your strategy in terms of raising that kind of money? And where do you see, like, how much more are you planning to raise? Of course, there is no end limit to what you can grow considering the field you are in. But tell us about the strategy first.
Yeah, I think especially when you're building a deep tech company, and in particular in biology, it's different from a lot of startups where you're kind of making revenue from day one. You can bring in revenue in a biotech company, and we've done that as well in terms of doing partnerships with pharma and biotech that want access to the technology. And that can be a very powerful source of non-dilutive funding. But in general, you have to raise a lot of money to build the technology, and then you kind of realize the profits of that a bit with like 5 to 10 years down the line.
Yeah, it's
which is very
delayed,
different
right?
from like a SaaS, like software business where you kind of are tracking your revenue month over month. No, like it's in some functional sense, often zero revenue and then billions of dollars of revenue conditional on successfully building the drug. At Mammoth, we're a bit of a unique company in that regard too, because we actually do have a lot of revenue from both doing partnerships in the therapeutic area, but even doing partnerships in other fields where CRISPR is useful, like diagnostics and agriculture, biomanufacturing. But kind of thinking more generally about deep tech, the most important thing is you have to find investors that believe in that long-term vision, because it's a very different journey from your typical SaaS startup. It's a journey where you have to have long-term capital. And that means that you have to really believe in the vision and believe that, hey, from the beginning of the company, it's going to be like, let's say, a 10-year journey. But at the end of that 10 years, you have the opportunity to truly build a $100 billion market cap company by actually curing these genetic diseases. And I think that these opportunities in biotech are surprisingly rare, because it's not often that you have this huge technology shift come along, like CRISPR, or in previous decades, RNAi, ASO, previous decades, antibodies. These are just rare shifts. So why we're excited also at Mammoth is we see CRISPR as one of these rare technology platform developments in biotech where you can actually build a $100 billion company. And the other thing that's unique is that biology is often this slog where you build one drug and it's just as hard to build the second drug and it's just as hard to build a third, because you don't have any kind of synergy between them. It's like, obviously you've learned something about building a company and things like that.
So is it really difficult? Like, you have to keep upping your game, especially when you go into the investors and, when you pitch for your product. Is it really difficult because you always have to seem better than what you were before?
No, I think in general, appropriately, the bar is always raising. And I think that's a good thing, right? It holds you accountable. And every time you go out for another round of financing, you need to show what progress you have made with, the substantial money that you've already brought in. And you have to be creating value out of that. I think to that point, one of the unique things about CRISPR that we're excited about at Mammoth is that it's a uniquely capital efficient technology. Because normally in biotech, building one drug and building the second drug takes just as much capital, takes just as much time, just as much scientific risk, basically. And one of the unique things about CRISPR is building the first therapy is the hardest. And then the second one's easier, the third one's easier, the first one's easier. Because for all of these therapies across all these like Alzheimer's, Parkinson's, Huntington's, you're using the same core protein, the same core CRISPR system, and you're just switching out one component called a guide RNA that's very easy to design and switch out. And that means that actually, once you kind of have that system locked, it's very easy to go from one disease to the next, to the next, to the next. And that's a very kind of new concept to biology, more similar to like a tech company. And that's one of the things that's really exciting and really makes us confident that we can build not just one or two drugs, but like 10, 20, 100 drugs using the technology and really build a $100 billion company, is that there is that kind of synergy between all the different drugs that we're building.
On one side, you are someone who is in the science and in the development side of things. On the other side, you are involved in fundraising and the business aspect of things. How difficult is it, and how do you switch between these two hats?
Yeah, well, first of all, I'm very fortunate to have a lot of great co-founders and executives who take on the brunt of pretty much all of these areas in many ways. But one of the pieces of advice that I got early on from a mentor was that when you're in the CEO role, you really have three jobs and you can rotate between them in various ways. But the three jobs are A, make sure the company's generally working on the right things. B, hire the best people in the world to help you work on those right things. And then C, don't run out of money while you do the previous two things.
Okay.
And the third one being maybe the most important in some ways.
Yeah, don't run out of money.
Financing and really kind of fundamentally storytelling, I think, is one of the most critical skills. And when I'm giving people advice who are kind of just starting out and founding their own startups or thinking about it, I think, especially for the CEO position, the number one thing is you have to love storytelling and you have to actually get energy out of kind of talking about the company, because that is going to be the number one job, whether that's external with investors or internal at the company or external with recruiting executives. Really, it's the fundamental thing that you're going to be constantly doing until, you're tired of saying the same thing 100 times. But you have to really, really enjoy that, because that, if you're shying away from that or you don't get energy from it, so you're trying to avoid it, that really is the fundamental job.
Would you use gene editing for cosmetic purposes and not just medical?
Yeah, I mean, it's definitely something you could do. I think the main question would be, have we proven the safety enough that that's something we want to do? I would personally be comfortable with it. I think that's a question for the society and the FDA in terms of, cosmetic applications. You can imagine something as trivial as like permanently changing eye color.
Because anti-aging, all of these things are such buzzwords, and people are ready to spend a lot of money for cosmetic purposes. And do you see that in the foreseeable future?
I think it's definitely something that's going to become increasingly technically possible because of innovations like what we've done at Mammoth in terms of, we can access the muscles. So, hey, what if you wanted to do a gene edit to have big muscles or something?
Yeah.
I think that's an interesting question in terms of, okay, what's the safety bar we need?
Because
it better be
really
1,000% safe before I think we should do that kind of thing. But the good news is so far the technology has shown to be extremely safe. So, we'll continue to study it. But I can totally imagine we live in a world where these are real choices that someone could make. Because it is an incredibly safe technology and we'll continue to develop it. And there are, genetic bases of things like muscle density that you could modify.
So a lot of CEOs, as we understand, I would love to know your take on it, because many of them decide that they do not want to spend a lot of time in the morning while getting ready, whether is it a black tea and I'm done with it, with that part of the brain processing that it requires, or would I like to invest a lot of time and make sure that, my outfit speaks the power that I wish to communicate? Which one of that, and what is fashion or dressing in that aspect mean to you?
Yeah, we can judge for yourself.
Well, you're quite impeccable.
This is a unique situation. I'm very much on
That's one-off situation.
I'm very envious of people that really can represent their personality through fashion. I think that's an incredible superpower, actually. But I'm very much on the other end of I try and simplify my life by basically wearing the same thing every day. Not a black t-shirt, but I have different colors. I have some flavor in my life.
Oh, thankfully.
But very much, I think that's an area where, due to a lack of education, I don't draw as much energy from. So I do try and just not think about it as much as possible.
So quickly we're coming to the end of our show. And I want to do a quick rapid fire with you. A book that changed your perspective?
Leaves of Grass by Thoreau. I think, yeah, there's phrases like the scent of my armpits finer than prayer. Just this idea that you can find beauty in the most
in the simplest things,
mundane and small things of life. I think it's a very powerful concept.
We're going to link that book for our audience. A scientist or entrepreneur you admire, you pay 2 hats, so which one takes precedence?
I mean, honestly, our co-founder, Jennifer Doudna, I mean, she is an incredible scientist, but also an incredibly, great human being. And those things aren't always aligned.
Together? Okay.
So I think that's a superpower as well.
The most unexpected challenge you faced as a CEO?
I think scientific training prepares you a lot for some of the hardest things in a startup, which are like dealing with uncertainty, dealing with no's, dealing with things that don't work, dealing with like not knowing. There's no answer in the back of the textbook, right? I think that's what it really prepares you for. What it doesn't prepare you for is like people management. And that's something that I do enjoy doing and find really interesting, but I definitely had zero training in it. And it's something where you just had to learn by doing. And I think that was something where also when I'm giving advice to people, I say like, really think about that. Make sure that's something you get energy from. Make sure it's something that you want to do, because aside from storytelling, probably the number two job. And that's not something a PhD really prepares you for.
Would you have the same answer if I asked you, what is a piece of advice that you wish you had received earlier?
Yeah, let's see, that I wish I had received earlier. I mean, I think a general one and this is really talking more to like scientific founders in particular, and like people coming out of a PhD or higher education. I think people are really self-limiting, truly. Like, I think often if you like kind of know too much, you start to in your head like say no to yourself. You're like, oh, like what if I try? Oh, but no, there's these 10 reasons it won't work. And that can be a really great trait in many ways, because you don't walk down the wrong path or you save some time. But I think it can be an incredibly devastating trait in terms of limiting yourself, in terms of like your career potential, or just in general really trying new things.
Overanalyzing.
Like way overanalyzing.
Overanalyzing, overthinking.
I think one of the biggest things is like really trying to have as much bias to action and letting others say no to you rather than saying no to yourself is a concept that I think a lot of people in academia and really coming out of PhDs, or at least I, let's say, without speaking for others, could have used much earlier in my career. I think that's a very powerful concept.
Okay, so one gadget or tech item you can't live without. Don't tell me phone.
I'm actually a bit of a Luddite. You can see I'm probably one of the only biotech founders that doesn't measure any. I don't have a Apple Watch.
Actually.
I
no
drink
watch,
tons of Coca-Cola.
Really?
I don't biohack. I probably do the opposite of biohacking. So my answer to this will be super boring. I have a really dumb Casio watch, and I only wear it when I'm running, and it's critical. I have it with me here. But that's really the only gadget aside from my phone that I use.
Okay. So are you a coffee or tea person?
Coca-Cola.
Oh
I was born in Atlanta, Georgia.
Somebody making the brand ambassador for it already.
Call me.
You know where to find me. Dr. Trevor Martin, this has been absolutely wonderful. Yep. Thank you very much. In fact, I would love to talk a lot more about it, because these are one of the topics that I personally enjoy, but unfortunately we're very, very short on time. We hope to have you back on the show sometime again soon. If you are in Dubai, please, you have to come back on the show with us, and we will explore a lot more other things that CRISPR is doing.
Cool. Yeah, no, thanks for having me, and hope the conference isn't too crazy for you. Keep watching Hustle, Style, and Stories with Shanu Suresh.